Alright, so we're going to start off our next section now looking at stems. Take a look at the overall organization of a plant stem, and then take a look at some specialized stems like we did in the last section where we ended with roots. As with roots, the other big organ system in the plant, the stems, contain three different types of plant tissues, right? So the plant tissues we know about now are vascular, ground, and dermal tissue. The stem is no exception. And just like in the roots, we're also going to have apical meristems, and even more so, we're going to see lateral meristems as well. The apical meristems then are going to produce tissue, and also typically at some point in time periodically produce what are called primordia. And primordia are what are going to be eventually leaves. So the apical meristem, this is a scanning electron micrograph. So if we see the scale here, this distance here represents 50 micrometers, right? So we're looking at 51 millionths or 5/100,000th of a meter. Very, very small. But this shoot apical meristem has produced a leaf primordium. This eventually, at some point in time, will become a leaf probably. There's a section here without that, and then another primordium, and a section without, another primordium, a section without, and so on, all the way down this very, very young stem. These then are the older primordia that are going to start to look like, if you were to zoom out from this picture by a few hundred times magnification, you would see what it looked like actual leaves surrounding the apical meristem and offering protection to it. Because remember, the apical meristem, the shoot, does not come with a cap like the root apical meristem does. There is no stem cap, if you will, like there is a root cap down below ground. So these primordia typically are going to turn into leaves eventually, but sometimes they can turn into shoots if you have a plant that produces a branching form, or even flowers if you're looking at a flowering plant. We've seen some of these terms before, but just to kind of orient you here, on the stem we have nodes, and so any point along the stem where something branches off, we call that a node. We can also look at internodes, so the distance between two nodes is called an internode, right? So there's a node here and a node here, and so the distance between the two is called an internode. We can look at a blade, which is the overall flattened part of the leaf, so this would be a leaf blade. We can also look at what's called a petiole, which is the part of the leaf that attaches it to the stem. So this kind of stalk right here that attaches the blade of the leaf to the stem, this part right here is called a petiole. The axil of the stem is the angle between the petiole and the stem itself. So here we have the petiole branching off, we have the stem going up. This angle between the petiole and stem is called the axil. And of course, periodically we do find axillary buds, which occur in the axils, which could eventually lead to, again, either another leaf or maybe even a secondary shoot of some sort. The terminal bud is the one all the way at the top, and typically the function of the terminal bud is going to be to protect the shoot apical meristem from herbivory, but especially also during wintertime, protect it from cold, because a really hard frost or really cold temperatures could actually damage the apical meristem, and if that's the case, that plant's going to have a really hard time growing down the road. Now, back in the root section, we also talked about monocots and dicots. Another difference between monocots and dicots is the arrangement of vascular tissue in their stems. So up here on the upper right, we have this section, a cross-section through what would be the stem of something like corn. Corn is a monocot, and monocots have their vascular tissues arranged in a sort of a random scattered fashion inside the stem. So there's really no rhyme or reason to how these little vascular bundles are arranged inside this stem. What you can tell is that within each one of these vascular bundles, inside of here, there's a bigger circle, which is going to be a xylem, and these two smaller circles are going to be the phloem cells inside that vascular bundle. But there is no like regular repeating arrangement of vascular bundles inside that stem. On the other hand, if you were to look at a dicot, what you would find is vascular bundles arranged in a ring. So if we take a look at this picture, there is a pith on the inside. Inside the ring is the pith. We then have xylem cells, which are kind of the bluish colored cells right here. These are xylem cells. We have phloem cells outside of that, which are the kind of reddish colored cells in this picture. And then separating those two would be typically something like a vascular cambium if this is a plant that does secondary growth. But even for a plant that doesn't do secondary growth, it is going to have a ring arrangement of vascular tissue in the stem. So that's a really key difference between monocots and dicots. All right, so we can see in the stem in this picture the beginnings of secondary growth. This picture up here is a stem during its first year of growth. So we have a pith, we have primary xylem, and we have primary phloem outside of that. Eventually what's going to happen is between the phloem and xylem, there is going to form a ring of cells going all the way through these gaps here between xylem and phloem, called the vascular cambium. It's going to go all the way around the stem. That vascular cambium, as it divides, as we see here in this picture, this is after a second year of growth in the same plant, we still see our primary xylem, which are the darker blue ones. We see primary phloem, which are the darker red ones. We can see our vascular cambium, which is the green ring going around the inside of the stem. And then just inside of that is secondary xylem. Just outside of that, if the lighter pink color here, is secondary phloem. And so what that does is that for every year the plant is alive, it produces a new ring of secondary xylem and a bit of new secondary phloem as well. So it's producing new vascular tissue every year, because every year what also happens is that vascular tissue loses some of its function. The older secondary phloem stop working as well or stop working completely, and so if this plant's going to stay alive and get bigger, it's going to have to produce some new vascular tissue. And so the tree rings that you think of when you think of a stem of a tree that's cut down, the rings that you can count are actually rings of annual growth of secondary xylem. Monocots don't do this. Monocots, for the most part, do not have any secondary growth. Just about the only exception to that would be like palm trees, because palm trees are monocots, but they do have a woody stem. But otherwise, monocots never ever do any secondary growth. This is that same plant now after, let's see, we've got year 1, year 2, year 3, year 4. This is after 5 years of growth. So we've got the original primary xylem, the pith inside of here. The original primary phloem is the dark layer of red. And then we've got these successive, the second year of growth, third year of growth, fourth year of growth, fifth year of growth tree rings. These are all rings or layers of secondary xylem produced by this vascular cambium. Just outside of that, the lighter pink layer is made up of secondary phloem, which are of course transporting food throughout the body of the plant. So those are our growth rings. What you might notice out here as well, this layer of clearish colored tissue out here, this is ground tissue. This purple layer then would be most likely the way it's shown in this picture, it's probably the cork cambium, which is the second lateral meristem, that produces what amounts to the bark of the plant. So that's what tree rings are. What eventually happens here is that inside of this stem, inside of this tree trunk that we're looking at here, the layers toward the inside that have stopped transporting stuff, they're no longer functional in terms of transporting water especially. They become part of what's called the heartwood of the tree. It's typically darker, it's got all kinds of chemicals in it to prevent it from rotting, but it's definitely not transporting water anymore. Just outside of that are the rest of the rings of secondary xylem called the sapwood. They typically are transporting water still at that point in time. And then we've got our vascular cambium. Just outside of that would be the secondary phloem. And just outside of that would be the cork cambium, which also produces the bark. Right? So the second of the lateral meristems is the cork cambium. Way out there in the cortex, that's going to start producing the bark of the plant. So the cork cambium, the cork it produces, and something called the phylloderm as well. Those are all part of what we typically would call the bark of the plant, which is going to help protect it from damage and protect it from the environment as well. Bark is also known as periderm. So if you see the term periderm, that is synonymous with the term bark. It's all the outer covering of the trunk in this case, in a tree. Stems also come with not only bark in some cases, but what are called lenticels. Lenticels are these, in this picture of the stem, these white spots. And what these are a means of gas exchange for the stem. Because remember, the stem is non-photosynthetic, so it's not making its own oxygen, but it still needs oxygen in order to carry out cellular respiration. So oxygen can occur through these lenticels. Here's a close-up view of one where we see CO2 going in, oxygen going out, or vice versa, depending on where you are in the plant. That's going to allow for gas exchange. So lenticels are also important. And most plants produce these or have these at some point during their life, depending on how old they are. You may or may not be able to see them, but they do typically come with lenticels to allow for gas exchange. Probably the place you're most familiar with lenticels would be like on an apple. You think about an apple, think about those little tiny white spots on the outside of the apple. Those technically are lenticels that are allowing for gas exchange with that fruit. As far as modified stems, we can look at a few different types here. We can look at what are called bulbs. Bulbs would be things like onions, flower bulbs as well. They are actually underground stems. Actually, I'm sorry, onions are not. These are stems. So flower bulbs, right? Those are modified stems that consist of leaves and then eventually below that adventitious roots. Corms are also similar to bulbs, but they don't have the leaves, and so some flowers will also grow through what's called a corm. And then rhizomes are a way for some plants to be able to spread throughout their environment horizontally. So in this case, we have a below ground horizontal stem moving throughout the environment, and wherever it finds conditions it likes, it sends down roots over here and sends a stem up over here, and then this thing is going to continue on its merry way until it finds a new spot to put down roots. We can also look at runners, which are above ground stems involved, kind of like an above ground rhizome. A runner is like an above ground rhizome, so strawberries produce those. Tendrils are modified stems as well. We can look at stolons, which are storage structures, and tubers, which are storage structures below ground as well. We can look at cladophylls. In a cactus, the big green part here is a stem. This is not a leaf, this is a stem. It looks like a leaf, but it's not. And the leaves in this case are the spines. So in this case, the leaves have been modified into a protective function to help protect the cladophylls against herbivory. Next up, we're going to take a look at leaves. And so leaves are produced then, of course, by leaf primordia, as we saw previously. This is, for the most part, where photosynthesis takes place, although some plants do have photosynthetic stems. For the most part, leaves are the main site of photosynthesis. Once the cells are produced through cell division, their job is to grow and eventually take on the appearance of the characteristics you would associate with a plant in terms of their leaf structure. Leaves are typically determinate in structure, which means they're going to get to a certain size and a certain shape, depending on the species, depending on conditions, and depending on where they occur on the plant. They're almost always going to get to a very predictable size and shape. And so we'll see different patterns of leaf arrangement. This one here is called an alternate leaf arrangement. If you were to look at an oak tree or something like that, the leaves are alternate. In other words, for alternate arrangement, there is only one leaf per node. So here and here and here, there's one leaf per node, and the leaves kind of bounce back and forth to either side of the stem, which prevents them from shading each other out quite so much. With opposite leaf arrangement, we see leaves on either side of the stem, 2 leaves per node in this case, on opposite sides of the stem, and as you kind of get a sense here, they sort of rotate around the stem as well to help prevent shading. And then a whorled leaf structure would be where you have more than 2 leaves per node, like in this plant over here. This would be like a bedstraw plant, for example. It's got a whorled leaf arrangement with multiple leaves per stem. There are different kinds of leaves as well, evolutionarily speaking and phylogenetically speaking. Microphyls are leaves that are very tiny, hence the name microphyll. They only have a single vein, and that vein doesn't even go the full length of the leaf. So these are what you find in very, very primitive seedless vascular plants like the one on this picture. This is called a club moss. It's related to ferns. It looks like a little tiny pine tree, but it's not even close to being a pine tree. The leaves on this organism here are very, very tiny. They only have that single vein, and so they would be microphylls. Things in the phylum Lycophida, which include the club mosses, would be mainly the ones that have microphylls. One of the first groups of organisms to have leaves of any kind. Megaphylls are the leaves that most of us are more familiar with. They are like the leaves of an oak tree or a maple tree, where you've got multiple veins throughout the leaf in various arrangements, transporting more things more quickly. That would be more of a megaphyll type characteristic. All right, so most dicots have, again, a petiole, which is the stalk that attaches the blade of the leaf to the stem. The flattening of the leaf allows the leaf to turn, especially when it's windy or breezy. The leaf can kind of move back and forth, and it helps to create circulation of air around the leaf to maximize CO2 uptake. Flattening of the leaf also maximizes absorption, so we have maximum sunlight absorption, maximum CO2 absorption. The downside of that is you also have maximum water loss, which is important in some ways, but not always in a good way. So we see a shift going from radial symmetry of leaves to what's called dorsoventral similarity of leaves. So in this case, we would actually, this leaf here would be dorsoventral symmetry. There's a single line of symmetry going down through the middle of the leaf this way. Radial symmetry would move more like a round leaf with no other features aside from that, really. It would make it more of a radially symmetrical leaf. Some leaves can have what are called stipules, which are little tiny fleshy projections that stick out of the petiole. A lot of the things in the rose family have stipules. They can also be spines, like we saw in the cactus plant. Those are modified leaves. The veins that occur in these things, the veins are made up of vascular bundles of both xylem and phloem. If you look at the veins of a monocot leaf, those veins are going to be parallel. They never cross each other. So the bottom picture here would be a picture of a monocot leaf with veins that never cross each other, versus a dicot leaf where you have what's described as more of a branched or netted network of veins throughout that leaf where they're kind of going in all directions. Again, another difference between monocots and dicots. We can also look at simple versus compound leaves. So in the picture up here on the left, this is a white oak branch. There are what we describe as being simple leaves. Each one of these whole structures here is one individual leaf. So here's one, there's one, there's one, and there's one. So those are simple leaves, undivided blades, one large contiguous structure that can have teeth on it, can have serrations on it, could have lobes on it like these oak leaves do, but they're not completely divided up, are the blades. Compound leaves are where you have the blades divided into leaflets. So this picture here would be like a sumac or something like that, where this entire big long thing here and this big long thing here, this entire thing is a leaf. Okay, it's what we call a pinnately compound leaf. This picture over here is of a vine called Virginia creeper, which we have in this part of the world as well. In this case, this entire ring of leaflets around the center part, this entire thing is considered a leaf. Right? And so the way you tell this apart is you look at the buds, right? So what you do is you'd find a leaf, you'd follow it all the way back until you see a bud, and from that point on, everything beyond that bud is a leaf. Okay, so that's kind of how you would distinguish between compound and simple. All right, so we're going to cut off here, and I will start up the next section here in a moment.